Hey Daisy, since you're starting a biology channel, I was wondering if you could make a video about photosynthesis?
Oh, absolutely, yeah.
That's awesome, because I've always wanted to know how it works.
Aren't you in AP Bio? Isn't your exam
Okay,
next week?
bye!
I won't sugarcoat it. Photosynthesis is kinda tough. You got big picture stuff and specifics that you need to know. But it's nothing we can't handle, so let's break it down. Alright, to start with big picture stuff. Photosynthesis occurs within the chloroplast. It consists of two processes. You have the light reactions and the Calvin cycle. The light reactions need photons of sunlight to occur, and the Calvin cycle doesn't depend on sunlight at all. In the light reactions, photons of light are absorbed by the chloroplast to make ATP and NADPH. That ATP and NADPH is used in the Calvin cycle, along with carbon dioxide, to produce glucose and oxygen. The formula for photosynthesis is something that you need to drill into your head forever, and is as follows. 6 CO2 plus 6 H2O plus sunlight makes C6H12O6 plus 6 O2. In simpler terms, you have carbon dioxide plus water plus sunlight makes glucose and oxygen gas. Alright, now that we've looked at big picture stuff, let's get into specifics. So we'll start with the light reactions. Zoom into the chloroplast, which we know is an organelle found in plant cells and cells of other photosynthetic organisms. Within the chloroplast, there are these discs stacked on top of each other like pancakes. These discs are the thylakoids. The whole stack together is called the granum, and the fluid within the chloroplast membranes is called the stroma. The fluid within the thylakoids is called the lumen. Zoom back into the thylakoid, specifically the thylakoid membrane. So let's take a look at this setup here. Along the membrane, we see these two globs called photosystem 2 and photosystem 1, which are basically proteins with chlorophyll inside. Photosystem 2 comes before photosystem 1, and they're only named that way because photosystem 1 was discovered first, which isn't confusing at all. We also see these proton pump proteins. Over here we have NADP reductase, which is an enzyme, and all the way at the end we have ATP synthase, another enzyme. This whole system is called the electron transport chain, ETC for short. So photons of light come in and are absorbed by the chlorophyll in photosystem 2. Chlorophyll is a green pigment, remember, so it's going to be absorbing red and blue light but reflecting green light, which is why leaves are green. The photons of light excite electrons in the photosystem, causing them to move super fast and eventually move out of the photosystem and onto the ETC. As this happens, two H2O molecules bind to an enzyme on the bottom of photosystem 2 down here. The enzyme breaks the two water molecules into 4 electrons, 4 protons, and 1 O2 molecule. The electrons replace the ones that leave the photosystem, the protons stay inside the lumen, and the oxygen molecules are released as a byproduct. Back to the electrons. The electrons leave photosystem 2 and travel down the thylakoid membrane, passing through and activating proton pump proteins along the way. These proton pumps take protons from the stroma and bring them against the concentration gradient into the lumen. At this point, there are way more protons inside the thylakoid than there are outside, so the only way for them to balance themselves out is if they go back through the ATP synthase down here, down the concentration gradient, where they can return to the stroma. Going through the ATP synthase causes ADP to phosphorylate with phosphate, and that creates ATP. But what about photosystem 1? Photons of light are also hitting photosystem 1, and once the electrons are excited there, they are snatched up by NADP reductase and combined with NADP plus to make NADPH. So that's the light reactions. Water plus photons of light makes ATP and NADPH, which are now hanging out in the stroma, right outside the thylakoid membrane. Now it's time for the Calvin cycle. So we're in the stroma of the chloroplast, and the ingredients we're going to need are ATP, NADPH, and CO2. We have the ATP and NADPH from the light reactions, and the CO2 is going to come through the stomata of the leaf. We're also going to need a 5-carbon molecule called RUBP. Its real name is ribulose bisphosphate, but nobody has time for that. Our last ingredient is an enzyme called Rubisco, which sounds like it should be a gas company in the Pixar movie Cars. The first step of the Calvin cycle is called carbon fixation. The first order of business is that the rubisco is going to combine an RUBP molecule with a CO2 molecule to get a 6-carbon molecule, because CO2 has 1 carbon, RUBP has 5, and 5 plus 1 equals 6. Ka-chow. All right. Oxygen is going to be released because we don't need it for the rest of this now, so bye-bye. The second step of the Calvin cycle is called reduction. This is where we're going to need our trusty ATP and NADPH. The 6-carbon molecule is split into 2 3-carbon molecules, and along come 2 molecules of ATP to give away their phosphates and become ADP, and along come 2 molecules of NADPH, which become NADP+ when they give away their protons. The resulting ADP and NADP+ are then recycled to be used in the light reactions once more. So one of the 3-carbon molecules that has been added to becomes what's known as pGal, because nobody is brave enough to say 3-phosphoglyceraldehyde. And quite frankly, we just don't have the time. Our pGal molecule is going to make its exit down here because that is all it needs from the Calvin cycle. The other 3-carbon molecule wants to become RUBP. This process is called regeneration. So you're probably thinking, Daisy, RUBP has 5 carbon atoms. How can this 3-carbon molecule become that? Well, down here it's going to get 1 more ATP, which becomes ADP, and then it's going to go through the Calvin cycle again 2 more times. Each time it goes through, the rubisco adds another carbon atom from the CO2 that's been cycling in. Something that also happens is that it gains 2 more ATP molecules and 2 more NADPH molecules over here each time, plus 1 more ATP over here that happens twice. And in the end, we are left with an RUBP molecule with 5 carbons. So that part's not too hard, but where I always get tripped up is in the math of things, so let's review. To make 1 pGal, you need 3 CO2, 9 ATP, and 6 NADPH. So it cycles through 3 times with the 3 CO2 molecules to make 1 pGal, 9 ADP, and 6 NADP+. And you need 2 pGals to make 1 glucose molecule. And let's not forget the O2 that's also released along the way. And that's the Calvin cycle. We started with 3 CO2 molecules and ended up with 1 pGal and some leftover oxygen. So to recap photosynthesis, let's revisit this formula. The H2O and sunlight were used in the light reactions in the thylakoid membrane to make ATP and NADPH and release some oxygen. And the CO2 plus the ATP and NADPH were used in the Calvin cycle, which takes place in the stroma, to make glucose and release some more oxygen. It took me a long time before I could fully grasp the concept of photosynthesis with all the different components and pieces of it and the process itself, so don't be too hard on yourself if you don't get it the first time. My advice to you is just to take some markers and some paper and to just draw it out a bunch of times until you really get it, just to memorize it. And that's the best piece of advice I can give you for this. Alright. Hopefully this video has been informative and helpful. I'm going to link some extra resources, including Mr. Birch's biotextbook, in the description of the video. And special thanks to Devin, my friend, for sending me the video from the Zoom call in the beginning. And an apology to him, because I didn't tell him what I'd be saying, I only told him his lines. I didn't mean to slander his name, so I want to make it clear that he does understand what photosynthesis is, and I did him dirty. Alright, thanks for watching.